Hello everyone, my name is Janie Brandsma, and in this presentation I will be discussing the bubonic plague, which is also known as the Black Death. Rodents are the primary host for the bacteria Yersinia pestis. Yersinia pestis survives in a zoonotic cycle between rodents and fleas. Plague is often associated with just rats, but all rodents including squirrels, chipmunks, and rabbits can contract the plague. A flea will bite an infected rodent, causing Yersinia pestis to enter the flea's digestive tract. Yersinia pestis will replicate inside the flea, causing a blockage of the digestive tract. Once the flea bites another organism, it will regurgitate and deposit the bacteria into the new host. Humans are considered an accidental host for Yersinia pestis when bitten by an infected flea. Plague has three different types it can appear as. The bubonic plague will develop simply by a flea bite, but it can develop into more lethal forms if left untreated. The septicemic plague is the most lethal form, caused by an overgrowth of Yersinia pestis in the bloodstream, which causes bacteremia. The pneumonic plague will develop when infectious droplets are inhaled. In this presentation, I will be focusing on the bubonic form of the plague and its effects on the immune system and the lymphatic system. Yersinia pestis will incubate in its host for up to 8 days before any symptoms will show. At the start of the onset of symptoms, there will be fever, headache, and weakness. It will rapidly increase in severity with nausea, vomiting, delirium, and giddiness as symptoms. At its most severe, gangrene will occur due to the loss of blood into tissue. The most notable sign of the bubonic plague is the formation of buboes. Buboes are the swelling of lymph nodes underneath the skin that are filled with either blood and/or pus. Buboes can burst, which is one way this disease can spread. The bubonic plague specifically can be diagnosed pretty quickly by the distinct buboes. With other forms of the plague, laboratory testing is required because symptoms may be similar to other diseases like dengue fever or pneumonia. Plague is tested for by collecting a sample from the patient. Blood, lymph fluid, a segment of a lymph node, or sputum are taken and then stained using either gram stain, Wright stain, Wason stain, or Giemsa staining techniques. Once the samples are stained and ready for viewing, a safety pin appearing bacteria will be a giveaway that this is a Yersinia pestis infection. On the image on the right, you can see how the bacteria has this kind of safety pin-like appearance. So a little bit about the bacteria Yersinia pestis. It is a gram-negative coccobacillus. This means that it has a thin layer of peptidoglycan that is surrounded by two membranes. Because of the double membranes, this bacteria can be hard to treat with antibiotics. It is encapsulated with polysaccharides, which can also make it difficult to treat. The capsule also aids in adhesion and evading the host organism's immune system. It is a facultative aerobe, so it can grow in either aerobic or anaerobic conditions. When cultured on sheep's blood, agar media, this bacteria will form gray colonies, which just look gross. It has a bipolar staining pattern, which is why it has that safety pin-like appearance. While this disease does have a history with killing millions of people in the past, it is not that common of a disease nowadays. There have only been around 1,000 cases in the U.S. within the last century. The bubonic plague makes up about 3/4 of these cases. This disease is most commonly acquired in the summertime due to people being outdoors more often. Rural areas contain the greatest number of cases, particularly in the southwestern states like New Mexico, Arizona, Colorado, and California. However, urban areas with high rat populations have also had outbreaks in the past. In 1925, there was a plague outbreak in Los Angeles that killed around 15 people. Because of modern medicine and public health standards, this disease is very uncommon. It will be impossible to completely eliminate this disease because of its zoonotic cycle. The only way to do this would be by exterminating all rodents, which would cause a massive impact on the ecosystem. The immune system is a complex set of organs that work together to protect the body from foreign invaders like bacteria, viruses, and pollen. All immune cells will be formed in the red bone marrow of long bones, such as the femur. The plague will negatively impact the innate immune system, which is the first line of defense for the body. It has a nonspecific response with the use of macrophages, neutrophils, dendritic cells, along with other white blood cells. Macrophages will kill and remove invaders by phagocytosing them. They will then stimulate the action of other immune cells in the adaptive immune system. The adaptive immune system is a specific learned response. Cytotoxic T cells of the adaptive immune system will kill certain cells, including foreign cells, viruses, and cancerous cells. Plasma cells are matured B cells that are able to produce specific antibodies in response to a pathogen. The lymphatic system is one part of the immune system. It contains several organs that include the thymus, liver, spleen, and lymph nodes. It will collect plasma from the tissues, turning it into lymph fluid. It will then return the lymph fluid back to the blood, which helps to maintain a proper blood volume and pressure. Before returning to the blood, lymph fluid will be filtered by the lymph nodes. Lymph nodes are commonly found in the neck, chest, armpits, and groin. The lymph nodes also store many immune lymphocytes, such as antibody-producing B cells. It will produce, store, and release lymphocytes as needed by the body. Once Yersinia pestis is inside the new host, it will quickly travel through the lymphatic system. It is unable to reproduce without being inside a cell because it is an obligate intracellular parasite. It will often choose macrophages to reproduce in. This will lead to a decrease in immunity. Because the immune system is suppressed, the bacteria will colonize the lymph nodes, causing swelling and tenderness. This is what causes the buboes to form. By this point, the host is usually too far gone, and the adaptive immune system will be unable to take effect. Yersinia pestis produces 6 effector proteins called Yersinia outer proteins, or YOPs. They are injected into a cell with the use of a type 3 secretion system. YOPs will block the immune system in several ways, including preventing the macrophages from doing phagocytosis, causing apoptosis of macrophage cells, and blocking cytokines from being released. The type 3 secretion system is a molecular machine found only in gram-negative bacteria. It acts as a hypodermic needle pumping the target cell full of toxins. The type 3 secretion system is encoded by one of 3 plasmids found in the cytoplasm of Yersinia pestis, specifically the plasmid PCD1. The type 3 secretion system is made up of several proteins. Some important ones include the LCRV protein, a YOP-B and D complex, and the YSCF injectosome. The LCRV protein found on the type 3 secretion system acts as the tip of the needle, allowing penetration into the target cell. It works in conjunction with the YOPs-B and D to allow the effector proteins to be injected. LCRV will also be released into the cell to help suppress the immune system. It will trigger the target cell to go into overdrive and produce around 40 times more interleukin-10 to be released. It also prevents tumor necrosis factor to be secreted from the macrophages. On the right, I have included an image of the crystal structure of the LCRV protein. It is composed of 322 amino acids in total. YOPs-B and D are hydrophobic proteins that are encoded by an operon found inside Yersinia pestis. They form a complex that will protrude from the LCRV on the type 3 secretion system that will form a pore for easy transport of other YOP proteins through the target cell membrane. The YSCD chaperone protein aids in the stabilization of the YOP-BD complex, along with efficient transport through the LCRV protein. Unfortunately, I was unable to find the crystal structure of YOP-B or D, but I have included an image of the YSCD chaperone protein found in the type 3 secretion system. It is composed of 347 amino acids in total. YOP-T is one of the toxic proteins that are created by Yersinia pestis. One of the ways that this protein causes harm to the host is by inhibiting phagocytosis. It does this by disrupting the actin cytoskeleton found in cells, which will prevent the macrophage from changing its shape in order to phagocytose the invader. YOP-T will also detach Rho GTPase from the plasma membrane, halting downstream signal pathways. Rho GTPase functions include regulating the cytoskeleton, cytokinesis, and vesicle trafficking through the membrane. So when it is detached from the membrane, none of these functions are possible. Many believe that YOP-T might be redundant or even unnecessary when in the presence of YOP-E. On the right, I've included an image of the crystal structure of YOP-T. It is a homodimer composed of 322 amino acids total. YOP-E, as I said before, has a very similar function to YOP-T. YOP-E will inhibit phagocytosis by macrophages and neutrophils similar to how YOP-T does. The difference is that instead of detaching Rho GTPase from the membrane, it will bind to it. This converts the GTPase into an inactive GDP-bound state. This will block downstream signaling pathways, preventing cytokinesis, vesicular trafficking through the membrane, and regulation of the cytoskeleton. On the right is an image of the homodimer YOP-E. It is composed of 219 amino acids, so it is slightly smaller than YOP-T. YOP-H is a protein that has the function of a PTPase, and it is actually known as one of the most potent PTPases found. PTPases will hydrolyze phosphate groups from a tyrosine residue with a phosphate group attached. The catalytic domain of this protein is found on the C-terminus. On the N-terminus is the binding domain. This is where the protein will bind to and dephosphorylate the macrophage substrates, SKAP-HOM or FYB. This renders them unuseful. On the right is an image of the YOP-H protein. It is composed of 468 amino acids total. The protein YPKA, otherwise known as YOP-O, has the function of a kinase. Similar to all the previous effector proteins, YPKA inhibits phagocytosis. The C-terminus is where YPKA will bind to an actin filament. The kinase catalytic domain is found at the N-terminus. YPKA will be inactive until it binds to an actin filament at the C-terminus, where it will then phosphorylate the alpha subunit of the GTPase, thus turning the enzyme off. On the right is an image of the crystal structure of the YPKA protein. It is composed of 732 amino acids, being the biggest out of all of the YOP proteins. Currently, the main treatment for the plague is with the use of antibiotics. Generally, streptomycin or gentamicin are used, but in the case of an allergy to these antibiotics, doxycycline or ciprofloxacin can be used. There are no FDA-approved vaccines yet for the plague, but scientists are readily working on developing one because there is a concern about Yersinia pestis being used in a bioterrorist attack. Killed whole vaccines have been attempted, but they are often too reactogenic, causing harmful side effects. This vaccine was attempted during the Vietnam War, but results showed that it was not a long-lasting vaccine. Live attenuated vaccines composed of Yersinia lipoproteins have been tested, but the results were too inconsistent to be approved. Subunit vaccines using the LCRV protein or the F1 antigen from Yersinia pestis have also been tested, and they are effective against Yersinia pestis, but they suppress the immune system too much, causing more harm than good. Like with many microbes, there are beneficial uses that come from Yersinia pestis. YOP proteins have been shown to help stop autoinflammatory flare-ups in diseases like Crohn's, irritable bowel syndrome, ulcerative colitis, or rheumatoid arthritis. The YOP proteins can help with autoimmune diseases by preventing white blood cells from going to the site of autoinflammation. YOP proteins can also be used to treat certain cardiovascular diseases like arteriosclerosis and even erectile dysfunction. YOP proteins help this by targeting Rho-associated kinase proteins. To summarize what you have learned during this presentation, I would like for you to label this diagram of a type 3 secretion system. On the right, I have included the terms required. And here are the correct answers so you can check your work. Thank you for listening to my presentation about the bubonic plague.